Hi, welcome to Moreauknows. Today's molecule we're going to be talking about are enzymes. The specific enzyme is called catalase. And catalase is an enzyme that breaks down hydrogen peroxide, which is labeled right here as H2O2. Now, hydrogen peroxide can move into catalase and be broken down into water and oxygen. So this is broken down, and I'll do the chemical equation here, H2O2. And to make this balance, you have to put a 2 here and a 2 here to make this equation balance. And the reaction that is formed from here can be done without use of an enzyme, but it takes millions and millions of years. Now, how do I know this is an enzyme? Well, usually enzymes have ASC at the end of their name. You might remember using hydrogen peroxide as a kid to put on a cut or to maybe bleach your hair. That was very dilute hydrogen peroxide. This is actually a picture of someone who has touched about 30% hydrogen peroxide, and that can actually kill cells. So it's not good for our body, and our body will break down that hydrogen peroxide using catalase. Enzymes are important to break down molecules, and they need something to help them do it. So I'm drawing here a protein, and inside this protein we're going to have an active site in blue. So this area right in here is going to be its active site. Now it doesn't actually look like this, of course, because you all know I'm an amazing drawer, but this substrate will actually fit into this active site and break apart or build up depending on which enzyme it is. So for our example before, the substrate would be the hydrogen peroxide and the enzyme would be catalase. Okay? So this is very important for enzymes. So all enzymes have an active site and they break down or build up substrates. Enzymes are important for chemical reactions, and these all affect the rate of the reactions. And that's the most important thing about the enzyme, is that it lowers activation energy and speeds up a reaction. When I say that it lowers activation energy, what I mean is if we had reactants and products over time, so here we would have time, and here we would have the amount of energy needed to go from the reactants, let's say we start here, to the products. Usually we would need a lot of energy to make those reactants turn into products. But in blue I'm going to have, with enzymes, it's just going to take a small hump and it will react and make products. So this activation energy is the difference between not having an enzyme and having an enzyme. And that can be a great amount. Like with hydrogen peroxide, it would take billions of years for hydrogen peroxide to break down into water and oxygen. But with enzymes, they do it in about 40 billion per second, which is really, really fast. So when I'm talking about an active site, what am I actually meaning? Well, this molecule here the hydrogen peroxide has to get into this catalase. And so it kind of looks like a mess there, but really there's a nice little spot for the hydrogen peroxide to go. And so if we look down here, we're looking at an enzyme right here, and this would be the substrate. The active site is this spot, and I'll draw it in blue again, right here. Okay? This fits and it lines up with the bonds on the substrate. So the substrate fits right in there perfectly. And so what we have here is called an enzyme substrate complex. And that's where basically they're just both together. And so usually we get water to come out of this reaction to split. So we will have, it's an exothermic reaction, which means it gives off heat. If we're putting products together, then it's endothermic and it absorbs heat. So the substrate comes into the active site. We form an enzyme substrate complex. Okay, enzyme substrate complex. And then that breaks the bond between the molecules. Okay, and then the products will leave out of the active site separately as two separate molecules. In this lab that you're going to be doing, you'll see that there are other items that were used besides hydrogen peroxide. And so what we're going to see is that only hydrogen peroxide is going to be broken down by catalase. And the reason for that is because this substrate right here fits into this enzyme specifically. It's one for one. So it's kind of like this lock and key, where the substrate, okay, is going to be the key, and then here would be the enzyme. And so the substrate being the key would fit into the enzyme. So the lock and key model is kind of what you want to think about. Thank you for watching Moreauknows. Now you do.